Hello. Last lecture we finished with the main parts of the light microscope that is used in the laboratory. Then we talked about the scales that we can see that can be used to see the different structure of any organelle, even up till the atom. And what is the scale of the light microscope, and what is the scale of the electron microscope, and what the scale of the normal eye to see which type of organelle or an atom. We'll start today talking about the different types of light microscope. The first type is bright field microscope, and this bright field microscope normally it is used in the laboratory where light passes directly through the specimen unless the specimen is naturally pigmented or artificially. This type of microscope have a little contrast. The second type used for stained specimen, it is a bright field microscope, and in this case you stain the sample with different stains, and in case you want to stain you need to fix the cell or preserve the cell, so you need to kill the cells. That's one of the main difference between both of these microscopes, because you can see in the bright field unfixed cells. The third type of microscope is called phase contrast microscope. Normally scientists use this type of microscope in cell culture so they can see live cells. The main advantage of this microscope that it uses a variation of density within the specimen. This variation in density is amplified and enhanced in contrast in unstained cells, so it can be easily seen these unpigmented cells. The other type of microscope, one of the very expensive type of microscope, is confocal microscope. Generally, confocal microscope uses laser to scan for the specimen to see the different structure. So in this image of the confocal, you can see two images. The top image showing a conventional or standard fluorescence microscope. So this fluorescence microscope in which each part of the specimen is tagged or labeled by a fluorescence dye. So you can see that you have like a green, orange and yellow. The yellow parts represent the overlapping between the green fluorescence and the orange fluorescence. So you can see as well in the standard fluorescence microscope, you can see that the image looks blurry. So by the use of the confocal microscope, the image and using the laser, the software can eliminate all the out of focus light from the specimen, and you can see that the image generated from the confocal software with the microscope is a lot sharper. By the aid as well of a software called deconvolution software, you can see that the difference between the use of the deconvolution software can be shown in the image in the lower part. So the upper part, when you layered different layers of the fluorescence above each other using the standard fluorescence micrograph, you'll see image like a 3D. But when you use the deconvolution software, it will remove these blurry images, and you have a very good contrast because it removes the out of focus light. The other type of light microscope is the differential interference contrast. In this microscope, the density difference between the structure or organelles within the specimen are amplified, so the image appear almost 3D. The normal fluorescence microscope, you can see, as we said before, you can tag different structures or organelles inside the cell as a sample to see different fluorescence emitting. This you can see from the photo that nucleus emit a blue light, while the cytoskeleton of the cell, like actin and myosin, emits green light, while the mitochondria emits like orange color. In this fluorescence dye, once you tag these cells with fluorescence dye, once light or laser pass on these organelles, it emits the light. That's so we can capture this by a photo. The super resolution microscope, you can see the difference between the top image and the lower image. It is used in a structure that is very small in size or diameter, reaching around 40 nanometer in diameter. So you can see from the standard fluorescence at the top figure, you can see still the image is very blurry, but at the below image, at the bottom image, you can see how the image is focused, because they use the super resolution. They use a software that that scans different levels of the photo to get a sharper image. The types of, now we finished with the types of light microscope. We will have two types for electron microscope. The first one is the scanning electron microscope, and you can see from the photo, scanning electron microscope only have an image like a 3D image of the surface of the specimen. So you can see in this image, you can see this is the cilia of the tracheal cells. It looks like beating, okay? Tracheal cells in the windpipe. So only scanning electron microscope help to see the 3D structure. Normally the photos appear black and white, but using a specific software we can colorize the photos. For the transmission electron microscope, if you need to see the internal structure of these tracheal cilia, you can do a transmission electron microscope. So you can see in the photo, you can see the cilia have been cut either longitudinally or cross-section of the cilia. So basically the difference between the two microscopes is the scanning, it's the revealing of the outer structure of the specimen, while transmission is revealing the internal structure of the specimen. But keep in mind that most of the time the photos are black and white, and the software is used to colorize the photos. Now we are going to talk about how you calculate magnification from the photo. If the specimen is viewed with a 10x eyepiece and a 40x objective lens, then the total magnification will be 400. Organelles in cells are too small to be measured in millimeter, so normally we use a smaller unit, which is a micrometer, which around 10 to the power minus 6 meter. The law of the calculating the magnification equal the size of image over the size of object. So if you consider having a mitochondria that appears in the photo, so how we can calculate the magnification? The magnification from the ruler, it will be 35 millimeter. You need to multiply with 1000 to have the unit as a micrometer. When we divide on the size of the object, the size of the object which is given is 10 micrometer. You will have the magnification around 3500. In another way, you can calculate the size of object as well, dividing the size of image over the magnification. So you have given now the given now is the magnification, which around 100,000, and you have the size of object using the ruler from the photo. So you have a photo that have an organelle that when you measure the size using the ruler, it will be 20 millimeter, and the magnification is given as well 100,000. When you use the same equation, you reach that the actual size or the size of the actual of the object is 0.2 micrometer. So the law, we can summarize the law is I equal m. I is the size of the image, A is the actual size of the object, M is the magnification. Again, magnification equal image over actual length. It is the same law. So you need to exercise these two exercises and to see how to use this information in first exercise to calculate the magnification from the scale bar, and from the second size to calculate the y, the actual length of the y in micrometer.